Hi students, we were talking about the lesson Chromosomal basis of inheritance, under which we have completed something about linkage. Under the linkage we have talked about, that is, what is coupling and repulsion theory, cis arrangement of genes, and then what is called the trans arrangement of genes. Now we have to go for the two types of linkage. So if you are taking linkage, there are two types. One is called complete linkage. One is called complete linkage. Another one is called incomplete linkage. Now complete linkage was studied or discovered by C.V. Bridges in 1990. So he also proposed, you know that one, X/A ratio theory, what is called genic balance theory of sex determination. C.V. Bridges known for genic balance theory of sex determination in Drosophila. So complete linkage was studied by C.V. Bridges and incomplete linkage was studied by XII. So complete linkage was observed in the case of Drosophila, particularly what we call the male Drosophila. Complete linkage was observed in male Drosophila mostly, and incomplete linkage was observed by XII in maize plant. Okay. What is complete linkage? Suppose if the two linked genes, say an example, if the chances of two linked genes or if the chances of separation of two linked genes are not possible, those genes remain close together. So there is no chance of crossing over. So only the parallel combinations are observed. I repeat once again. If the chances of separation of two linked genes are not possible, I am taking here for examples capital or small l. They are present close together. They do not exhibit what is called crossing over. There is no chance of crossing over because the genes are closely linked together. So, if the chances of separation of the two closely linked genes are not possible, and those genes remain together, as a result, only we can receive the parental combinations. If crossing over occurs, then only we have what is called new combinations occur. But here in the case of complete linkage, the genes are found close together. There is no possibility of separation of chromosomes. There is no possibility of separation of genes. So that the genes remain close together. As a result, we have received only parallel combinations. So here I am taking for example capital or small l. These two genes are found close together. They do not exhibit the phenomenon of crossing over. Though these two genes are present on the same chromosome, as they are found close together, they do not exhibit the chances of crossing over. And this is called complete linkage. As I mentioned earlier, complete linkage was observed in male Drosophila. C.V. Bridges observed complete linkage in many species of Drosophila. So there is no separation of genes, no new combinations are found in the case of complete linkage. Now in the case of incomplete linkage, suppose if the two linked genes are placed far apart, I am taking this is a chromosome, it is placed here, far apart, here closely placed, that's why there is no chance of crossing over. But if the two genes are actually located far apart, away from each other, and if there is any chance of crossing over, so there is a chance of separation of what is called we have the genes because of crossing over. And this is called incomplete linkage. Okay, so if the two linked genes are placed far apart and their chances of actually separation is possible, here not possible, here possible, then the phenomenon is called incomplete linkage. We have received both parallel combinations and some new combinations. Here only parallel combinations in the case of complete linkage. Here both parallel and some new combinations are formed. So here there is no separation of genes possible. Here separation of genes possible. Here there is no crossing over, but here there is a crossing over. As a result, here we receive only the parallel combinations, but here because of chances of crossing over, we have received both the parallel combinations and new combinations. We will see in the example. So example for this one male Drosophila and some species of Drosophila, and for incomplete linkage that was observed by XII, that is in maize. That's about what is called complete linkage and incomplete linkage. We have to explain these two with examples. Example 1, example 2 with illustration. Now let us take the complete linkage in Drosophila. Let us describe how far the genes being together in the same chromosome, they are being inherited together. I mentioned already we have an example, the best example of complete linkage, the male Drosophila. So C.B. Bridges observed what we have actually complete linkage in many species, or we can say some species of Drosophila. And here, just he took two colors. Sorry, I made a mistake, two characters. One character related to the eye color, another character related to the what is called the shape of the wing. So eye color become many shades. Here he took two colors of eye. One red eye color, another one purple eye color. The genes for eye color are located in X chromosomes. Then he also took another pair of characters. One is vestigial wing, the one which is not fully developed, and another one the long wing, the one which is fully developed. So he took two individuals, homozygous individuals, one with the red eye and vestigial wing, another one with the purple eye and long wing. Now here the red eye is dominant over purple eye, and the vestigial wing is recessive to long wing. So long wing is dominant and the vestigial wing is recessive. So you see, from this one you can understand, it is an example for repulsion also. Because you see that one, the dominant gene is present in one chromosome and its recessive gene is present in another chromosome. Like that. Okay. So that's why it's also an example for repulsion. We don't want that one. We need to know whether the genes remain together or not, exhibiting complete linkage. So he crossed red eye, vestigial winged male with the purple eye, long winged female, because it's a sex-linked character located in the X chromosome. Now, these were the genes located. So we have, this is a genotype or recessive gene L and or dominant gene and recessive. You see the location. So in one chromosome we have dominant gene and a recessive allele for vestigial wing. Another chromosome we have dominant gene and the recessive allele for vestigial wing. Now here we have the recessive genes for what we have that is the purple eye color and the long wing for the dominant character. So as a rule, you know that one red eye is dominant over what is called just actually other eye colors, purple eye. Now the F1 generation we have received red eye color. And similarly about the inheritance of the wing size, the long wing is dominant over the vestigial wing. So we have received only the long wing in the F1 generation. So if you are taking the F1 individuals, now we have red eye with a long wing because of the presence of dominant letter R and the dominant letter capital L for long wing. So normally the gametes produced by the parallel varieties have capital R, small l, small r, capital L. These are the gametes produced by what we call the parents. By the union of these two gametes we have received what is called F1 generation with the red eye color with a long wing. This is because of the presence of one dominant gene is enough to produce a color or capital R and then one capital L. If you are taking the genotype of this one, you have the genotype. This is the genotype of this offspring. Capital R, small l and then small r, capital L. But we need to have one dominant gene enough to produce a character so that red eye and long wing being produced as a general rule. Now the next one, tested cross. The F1 heterozygous individuals were crossed with homozygous pure recessive parent, or we can say double recessive parent because it is a diagonal cross. So we can say double recessive parent. So the F1 heterozygous individuals are crossed with what we have actually double recessive parent. Here the F1 individuals are red eye with a long wing, and the double recessive parent, you know that one, purple eye, vestigial wing, because these two are the recessive characters. As we have two recessive characters, the parent is called double recessive parent. Now it is a test cross, crossing up F1 heterozygous individuals with what we have a double recessive parent. Now the gametes produced. So normally the F1 heterozygous individuals, as we studied already in the case of coupling and repulsion illustrations, the heterozygous individuals produce four types of gametes as a general rule, as seen already in the case of coupling and repulsion. But here, in this case, no four types of gametes form. Because the genes are linked together, they are tightly close together because they are present close together. There is no chance of crossing over as the genes are found very close together. I represent, you see that when the genes are found very close together. So there is no chance of crossing over. As a result, we are getting only two types of gametes. You see that one, this is one type of gamete, this is another type of gamete. One gamete with a capital R and then small l, another gamete with a small r and capital L. So if you have actually, if you would have just actually received, or if you would have, for example, the crossing over occurred, then we can say there is a possibility of getting four types of gametes. But here there is no chance of crossing over because the genes are actually close together. As a result, the F1 heterozygous individuals produce only two types of gametes instead of four. And whereas you know that one, the homozygous recessive parent produces only one type of gamete. So the possibility would be, you know that one, we are getting only just two different types of individuals. For example, this is one type of individuals, and this is another type of individuals. See, only two types of gametes on one side, there are only one type of gamete on the other side. So the probabilities are 1 into 2, just actually two types of individuals in the F2 generation. That is what we have. The red eye with the vestigial wing, the parallel combination. Another one, purple eye with the long wings, again the parallel combinations. So we took these two, only the parallel combinations. For example, red eye with the vestigial wing and purple eye with the long wing. So red eye vestigial wing and then purple eye long wing individuals are produced. These are the parallel combinations. So only parallel combinations are produced and no new combinations are produced because there is no chance of crossing over. So from this experiment, C.B. Bridges concluded that the genes for red eye color, the genes for vestigial wing, and similarly the genes for purple eye color and the genes for what we have the long wing are tightly linked together, so avoiding the chances of crossing over. So instead of getting the ratio 1:1:1:1, we are getting only 1:1 ratio only, and these two are only the parallel combinations. So it is observed in the case of male Drosophila and also some species of Drosophila also. Okay, so this is about what we have complete linkage. In complete linkage, there is no chance of getting new combinations. We have received only the parallel combination. This is because of the absence of crossing over. Okay, so that's about the ratio observed in the case of complete linkage 1:1 instead of 1:1:1:1. Now let's go to the next one, what we have incomplete linkage as observed in maize by Hutchinson. Now let's see incomplete linkage as seen in the case of maize plant. You know, in incomplete linkage, the two closely linked genes normally not there. We have the genes are placed fully apart or sufficiently apart, and their chances of separation is possible. So that we have received both parental and non-parental combinations, or we can say recombinant progenies will get it. Now here, Hutchinson took two characters just like a dihybrid cross. One is the color of the seed, another one the shape of the seed. One homozygous plant with both the dominant characters, that is colored full seed. The colored full seed is normally a dominant character. Colored one is a dominant character and full seed is a dominant character. The another one, the colorless seed, which is being shrunken, that is normally considered as a recessive character. So both the characters are recessive here, here both the characters are dominant. He crossed actually the homozygous individuals. As a general rule, you know that one in F1 generation, the dominant character is exhibited. Now in the F1 generation, we have received a character what is called normally colored full. Colored full. This is a F1 generation individual. These are the gametes formed. See here I represented the two genes, namely the gene for color, the gene for shape. They are being placed fully apart, sufficiently apart. They are not being close together. somehow they are placed away from one another. That's why represented the illustration. Now the F1 generation we are getting colorful. When this F1 heterozygous individual, that is a colored full seed, is normally crossed with a double recessive pair. You know the expected ratio. We are getting 1:1:1:1 in a recessive back cross, in a recessive test cross. But because of what we have some incomplete linkage, what will happen? There is a chance of getting new combinations also. We are not getting what is called the 1:1:1 ratio. We are getting what is called the coupling ratio. What we can say 7:1:1:7, like that. But somehow crossing over took place. So we are getting least number of recombinant varieties because you see that one actually crossing over occurred somehow, not fully. Not complete crossing over, not complete linkage, but we have some chances of crossing over. That's why we are getting two new varieties as in the case of test cross, but the ratio is not the same, just like 1:1:1:1. But we are getting a different ratio. Now the test cross result. As a general rule in a test cross or in a diabetic cross, we are getting what's called four types of individuals. One, the colored full seed, another one colored in the shrunken seed, and another one colorless full seed, and the last one colorless shrunken seed. Now we started the experiment with the colored full seed. There is a dominant character, the parent, another parent with both the recessive characters, colorless shrunken seed. So these two are called actually the parental combinations, parental progeny. So here you see that when the parental combinations are more in number, this is because of we have linkage is possible, but somehow crossing over took place. So that we are getting new combinations of what is called colored shrunken seed and colorless full seed. These two other recombinant varieties are new combinations, but they are least in number. Okay, so increased parental combinations, normally you can get it only in the case of coupling, 7:1:7, or we have in the repulsion, but here this is what we have the coupling, just or cis arrangement of genes, or we have due to some factors, we have received more number of parental combinations. So parental combinations are more in number, and we have actually the recombinations are least number. So due to crossing over, recombinant progenies obtained in least number. Though we are getting in least number, but crossing over occurred. That only we have received what we have new combinations. If there is no crossing over, definitely you can get only the two parental combinations as seen in the case of, for example, Drosophila. But here, because of crossing over between the genes, we have received new combinations also, but in least number. So this is about what we have the incomplete linkage as seen in the case of maize. So compare both what is called complete linkage in the case of Drosophila and incomplete linkage in the case of that is maize. There you have received only two combinations, the parallel combinations both. Here we are getting what is called actually four combinations, two combinations of parallel types and two combinations of recombinant types. Okay. Now what are linkage groups? The groups of linearly arranged linked genes on a chromosome are called linkage groups. Normally, one linkage group is equal to one haploid set of chromosomes. One linkage group, or we can say the number of linkage groups corresponds to the number of chromosomes in a haploid set. So number of linkage groups is equal to number of chromosomes in a haploid set. Now for example, in the case of Drosophila, we have 8 chromosomes. The haploid set is equal to 4. So number of chromosomes in diploid is 8, but the number of chromosomes in haploid is 4. Therefore, the number of linkage groups is equal to 4. So one linkage group is normally equal to one chromosome in a haploid set. Or we can say, in this manner, number of linkage groups is equal to number of chromosomes in a haploid set. If you have 8 chromosomes in a haploid set, we have 8 linkage groups. If you have 4 chromosomes in a haploid set 4. For example, in human beings, we have 23 linkage groups because we have in a haploid set 23 chromosomes are present. So in Mucor, we have 2 linkage groups, that means the total number of chromosomes 4. In Drosophila, I mentioned about haploid set 4, diploid 8. Now in sweet pea, there are about 14 chromosomes. In a haploid set, we have 7. So the number of linkage groups is equal to 7. Similar is the case in the red mold Neurospora. Here we have also 7 linkage groups as the total number of chromosomes in a diploid set is 14. Now in maize, the total number of chromosomes 20, haploid is equal to 10, so the number of linkage group is equal to 10. Then in humans, I mentioned about we have 46 chromosomes and haploid set is equal to 23. Therefore, there are about 23 linkage groups. Okay, so the number of linkage groups is equal to number of chromosomes in a haploid set. Okay, so that's about what are here the linkage groups. Now Mendel did not know 2 phenomena. One is linkage, another one crossing over. Now what are the differences between linkage and crossing over? These are the 2 phenomena which have opposite effects. One is doing one thing but the another one is doing it opposite way. Normally the linkage keeps the genes together, whereas now with the crossing over mixes them. So stay together actually keeps the genes close together, and what we have the crossing over mixes the genes. Now what are the main differences? Now here we see that one in the case of linkage, the linked genes present on the same chromosome stay close together. The linked genes present on the homologous chromosome stay close together. Whereas in the case of crossing over, it leads the separation of genes because of mechanism of crossing over. Then we have the second difference, linkage, it involves same chromosomes of homologous chromosomes, same chromosome. If you are taking for example the homologous pair, we have for example LL like that or LG, any genes. So it involves the same chromosomes of homologous chromosomes. They are present in the same chromosome of a homologous member. But here what is happening, we are going to see the mechanism. Exchange of chromosomal segments occurs between the non-sister chromatids of homologous chromosome. Exchange of chromosomal segments occurs between the non-sister chromatids of homologous chromosomes. Suppose this is one pair of chromosomes in what we have splitting stage, duplicated stage. This is another pair. These two are called actually the sister chromatids. The chromatids present in the same chromosome are called sister chromatids. Suppose we are comparing these two. Chromatids of 2 homologous chromosomes in a pair. They are called as a non-sister chromatids. These two are non-sister chromatids. So crossing over occurs only between the non-sister chromatids of homologous chromosomes. So exchange of chromosome segments between the non-sister chromatids of homologous chromosomes occur here. But here the genes present in the same chromosome are inherited together. Now the third one, what is its significance? It reduces new gene combinations. No new gene combinations are actually occurring except in the case of incomplete linkage. In complete linkage, complete absence of new what is called combinations. But in the case of incomplete linkage, we have some more crossing over occurs, so we have new combinations. Anyway, linkage reduces new gene combinations. But in the case of crossing over, because of crossing, it increases variability, differences. So new gene combinations occur, which leads to the formation of new organism. So variability increases, new gene combinations occur, which results in what we have formation of new organisms. So these are all some of the differences between linkage and crossing over as per the syllabus. Now we have to proceed the next one, the mechanism of crossing over in the next class. Okay, till then goodbye, thank you.